Okay, class. So this is going to be what we're going to call lesson 22, which is lecture 17, which is prokaryotic gene regulation and genetics of bacteria. Okay, so the basics of bacterial genetics is when you grow bacteria, you have bacteria in a broth, usually like it's a luria broth or something like that, and it provides them with the nutrients they need to survive. So sugars, essential amino acids, that sort of stuff. And you'll take that suspension of these bacteria in this broth, and then you can pipette it or loop it onto an agar surface. So this is made out of agarose, which is a sugar, and generally this contains all the things the bacteria need to survive. So sugars, um, lipids, um, you know, like basically it's ground up yeast and stuff like that. But we can vary the components that we add into the agar as well. It's kind of like Jello if you haven't seen it before. You make it the same way. You cook it, then you chill it. Instead of making like orange flavoring and you know table sugars, or fructose, um, it's generally ground up yeast with some sugar supplements in it. So anyway, you would spread the bacteria on the surface. After you wait a couple days, the individual bacteria will divide through mitosis, which we talked, well, not my binary fission, and then they continue to divide until you have millions of these, which produce a colony. So a colony is like a circular area. Theoretically, it's a clonal replica of a single bacteria that's grown on that plate. And of course, these are in petri dishes, um, and you cannot see the individual cells with your naked eye. You'd have to need a microscope. But after it incubates for a couple days, those individual colonies will turn into millions, and then you can see it with your naked eye. Just like think about a a piece of DNA, a nucleotide of DNA. You can't see that, but if you look at chromosomes uh, under the microscope with thousands and millions of nucleotides, you can see that. All right. So that's basically how bacterial genetics works. Okay. So um, when I was doing research, I started the biotech program here in 2004. And one of the things that I wanted to change was the labs being cookie cutter. So we partnered with people at Monsanto who had sequenced whole bacteria, 4 million nucleotides. Back in the day, that was no small feat. Today, it's not that big of a deal. But um, we wanted to see if we could figure out what each of those genes were. So we just had like millions of A, G, C's, and T's, and we wanted to have students introduce transposons, which we're not going to get to this semester. But basically, they're jumping genes and they can jump in and out. And so when they would jump in, let's say that this is a gene, um, let's say, there we go. Let's say this is a gene, and this gene codes for the ability for this bacteria to break down the sugar lactose. So we're going to call it a lac gene. And so what would happen is these transposable elements we would introduce to the bacteria, they would randomly insert themselves into these genes. And knowing what you know, I'm sure you know that this is not going to help make this particular protein, so it would knock out this gene. So we can make knockout genes and then we grow them on minimal media to see what's been knocked out. So it's kind of a cool, simple experiment. So you can figure out what genes are involved and what function um, by adding these transposable elements. So, a couple terms when you're doing this sort of stuff, these knockouts or whatever, or growing them on minimal media. When we talk about wild type bacteria, they're called prototrophic. All right, so that's just the word you're going to need to know. That means they grow and divide on minimal medium, which means it's the media, it's stuff in the agarose that the bacteria need. It's the minimum amount they need to survive. So salt, a carbon, energy source, water, etc. And that's what we refer to as minimal medium. So if, let's say, we knocked out a gene uh, that the bacteria needed to live, like, I don't know, we'll get to an example in a second, but we could supplement that, uh, whatever that is, into the media. Okay, so nutritional mutants, these are not wild type. That means they cannot grow without some sort of specific um, cellular building block, uh, or some sort of energy source. And this is the other term, so we call these auxotrophs. That means that something is mutated in them so that they cannot live on minimal media. It has to be supplemented with whatever they're missing, whatever's been knocked out. So no growth when one particular energy source is given. It can't utilize that source because maybe it doesn't have the genes to utilize that source. All right. So here are some genotypic symbols used in bacterial genetics. So and a lot of these things are in your problem set. So bio minus means that you have to add biotin into the minimal media because it can't make its own biotin. Uh, this arginine minus means that it can't make arginine. So you have to add arginine. This is an amino acid if you don't know, into the minimal media for the bacteria to live because it's an essential amino acid. It cannot function without arginine. It can make proteins. Met is methionine, so it needs that. Remember that's another amino acid that every protein ever made starts with. Lac minus means that it cannot utilize lactose as a carbon source. Gal minus means it can't use galactose. Um, STR means this R means it's resistant, so it's resistant to streptomycin. So if you add streptomycin, it wouldn't kill this bacteria because it's resistant. This S means that it's sensitive to streptomycin. So if you give the antibiotic streptomycin, it would kill this bacteria. So there's a lot of these. We're going to focus on these few. So just know these, but I mean, there could be like other things like azithromycin, and this is important in medical fields too. So, anyway. All right, so this is how we screen. This is how we would screen for our mutants when we were doing the transposon elements in the biotech program. Obviously, like if you have a thing, an organism, let's say that you're trying to knock out biotin. Right, so you would have to grow these on a lot of different supplemental minimal media plates. So this plate here, this primary plate, has minimal media and biotin, right? So let's say your goal is to knock out biotin. And so this is the biotin plus gene, and you put a transposon in there and it knocks it out. So it becomes biotin minus. Well, this thing can't make biotin, So it's not going to grow on a media that doesn't have biotin in it. You couldn't just go straight to minimal media because this colony would never show up. It would be dead. So we supplement the media with biotin, and then we're going to grow all these back. So we're going to grow ones that make their own biotin, ones that don't. It's not going to matter. So what we have to do is something called replica plating, and this is a simple procedure. Basically, you just get a piece of velvet or whatever. There's tools that they make, but you could do this on your own. And basically, you just push it onto the plate so it picks up colonies on it. And then it could be a filter paper or anything. You can't smear it around. So you just touch it to the primary plate and then you move it over to the secondary plate and you push it down. And it basically is replicating the same colonies that are on the original primary plate. So in this case, you would this is just minimal media, right? This is the secondary plate, no biotin. So anything that can't make biotin is going to die. So what you're looking for here is what's missing. What's missing from this plate that's present on this plate? Well, this one's there and these are there, and so all of these are the same, but the one that's missing is this one. And so because this one is missing, we can go back to the original plate and take a sample of this one, and then we can do our genetics on it, right? We can find out like where is the mutation in this biotin. You know, where did our transposon jump into it? What pathway is this gene involved? Are there other pathways that it can go around, and so on and so forth? So that's how we start studying bacterial genetics. Um, and it's done using this replica plating technique. All right. So scientists love to look at Escherichia coli, and the reason is because it's the most highly studied bacteria that exists. Um, it's super symbiotic in humans and other animal guts. So, like, it was actually named after a physician. His name was Escherich, and it's called coli because he found it and he discovered that every person's colon that he looked at had this bacteria. So it just symbiotically lives in colons of animals. It's a single circular chromosome. So, like most bacteria, we talked about this. Bacteria have circular genomes. Um, Other eukaryotes generally have linear one. Yeast is like this. So yeast and most bacteria, some bacteria have linear chromosomes, but it just, I mean, if I told you a rule, there's always going to be an exception to it. So, this is just a general knowledge. So we have the bacteria that are from the gut, the discovered by Escherich, uh, the physician, in people's colons. It's a singular chromosome. This is about the normal size, so about 4 megabases. Sorry. It's haploid, which means it only has one copy of its alleles, right? Not diploid like we are, and we already talked about that. It's unicellular, grows by cell division. It's asexual reproduction, although it can do conjugation. We're going to talk about that. And so some might consider that sexual. This is not meiosis or mitosis. It's a process called binary fission. It's not 100% completely understood how this works, um, but it would be a great uh, experiment project for someone who wanted to win a Nobel Prize, because if you could stop binary fission, then you could kill the bacteria because they wouldn't be able to divide. And, that would be a great antibiotic. Okay, so bacteria can transfer. So if we look at a bacteria, and they have a chromosome in it that they can transfer their genetic material. So it's called rolling circle replication. They build something called a pilus, or um, you can call it pilus if you want. I don't care. Um, so it's basically just a bridge between the two cytoplasms, and there are enzymes that can transfer the DNA. So it makes a copy of this haploid, and then it can transfer the DNA to a new bacteria. This is how they figured out that this happens. So scientists took, we'll just call it A. So this is methionine minus, biotin minus, threonine plus, leucine plus, um, this is thiamine. So threamine, thiamine. I just had to think about that for a second. Anyway, it doesn't really matter. Um, so these are gonna these are gonna get plated on a minimal media, right? They don't have methionine, they don't have biotin. Without these two being supplemented into the minimal media, they're gonna die. So the only colonies that are gonna, I mean, the artist is showing where they would have been, but these are basically dead. Nothing's gonna grow on there because it can't. Um, so I don't know what these red dots are, but they are what they are. This one is B. We'll call it just strain B. And so this is Met Plus and Bitin Plus, which is opposite this one. All right. And then threonine, leucine, and thiamine. Sorry. I don't know why I keep forgetting that. Thiamine um, minus. So again, opposite of this. So they mixed the B's and the A's. Of course, nothing's going to grow on this. This is minimal media. It can't make any of these amino acids. So they're going to be dead. This is a clean plate, even though it's clear. There's nothing going to grow in here. This one is also clear. Nothing's going to grow in there. They mixed them together. So mixing them together, you would expect nothing would grow, but you did get colonies, which is what the artist is showing these purple things. And when the colonies grew, you could take them out and look at their genetics and figure out that they're Met plus, Bio plus, 3D plus, 3D, and so on. And so these are now normal wild type prototrophic colonies. They have all of them of their important genes intact so that they can produce all of these products where they couldn't before. So, what the heck is going on here? These were transformed to pluses, right, from here, and these were transformed to pluses. So, how did that happen? And so, the way it happens is that scientists, looked at this and they found something called the fertility factor, which we're going to call F. F. So, certain E. coli strains, we're going to call these donor strains, so that they have the fertility factor. They're F plus. So here's the bacterial chromosome, and then there's exogenous pieces of DNA we call plasmids. So these are usually small, uh, you know, 4 to 20,000 bases. This is usually big. We're talking millions of bases. And so this donor strain, it's F plus, so it has a fertility factor, can create this pilus that we talked about, and it can transfer its. It's not going to be able to transfer its whole bacterial chromosome through this because that would take forever. But it is since this is so small, this is relatively easy to do. And so it can make a copy and send part of that into uh, an F minus. So that F minus would not have the fertility factor, and that would be the recipient. And that fertility factor is a gene, and it's usually located somewhere in this plasmid. So F pluses can donate their external exogenous plasmid. You need to know this name, DNA, using rolling circle replication, which is basically the circle rolls as it's replicated. So it's showing that here. This is single stranded right here, and then it becomes double stranded once it's in the donor or the recipient, sorry, not the donor, bacteria. All right. These plasmids replicate independently in the cytoplasm of the bacteria. Okay, so there are some there are some instances where the fertility factor, so this is an F plus bacteria, it has the gene for the fertility thing, which means it can transfer its DNA to another organism. And in this case, this plasmid can get incorporated into the chromosome. So this small plasmid gets incorporated into the chromosome, which is shown here, and now it's red. So it's integrated, the fertility factor, that gene is now integrated into the chromosome of the bacteria. And so we call this an HFR, which stands for high frequency recombinant. All right, so this is just basically telling me what I just said. Okay, so we can figure out the order of genes in some instances by looking at these high frequency recombinants. And so let's just say here's the fertility factor. It usually starts in the middle, so the transfer, the rolling circle replication usually starts in the middle of the fertility factor. So the DNA is cleaved, it transfers across this pilus into the recipient. Remember, the recipient has to be an F minus. So the donor can be an F plus or it can be HFR because it has to have the fertility factor either in its chromosome or in that plasmid. And that's the difference between F plus and HFR. So it cleaves it in the F and then it starts pushing its DNA, rolling the DNA over. So in this case, the A is going to go across first, which is showing it in this order. This is first. This is second, right? So we're going, if I took this and I pulled it across, A would be the first, B would be second, C would be third, right? And so this bacteria is now temporarily um, a, a diploid, right? It has two alleles for each of these traits. And in this case, these would be heterozygous. Plus B, C plus B plus A minus C minus B minus A. And this is going to continue until this gets disrupted. Remember, this is like 4 million nucleotides. To transfer an entire chromosome would take forever. And this recipient is not going to become an F plus because it starts in the middle of F. So it would have to go all the way around the entire 4 million bases back and complete the circle. And that would take out like hours. I don't know how many hours. I'm going to say 20 to 30 hours. And this pilus is not going to last that long. This is real. I mean, it's between two cells. It's super fragile. So usually, this thing breaks and whatever goes across goes across. So let's say, it had enough time, a few minutes to get A and B and C across. So it gets it across. Once that single strand is put across, uh, the enzymes in the bacteria build the second strand based on the first strand. And so in this case, this thing is diploid, right? So this is just telling you this is converted into a double helix. Now, there, like we talked about, we have crossover events. When these are near each other, we can cross them over. So in this case, this crossover would make B and A. So this would become a B plus, right? Because B is going to get transferred, A is going to get transferred, and then these would go out. C's not in the cross. C's outside of the cross, right? Here's the cross, right? Right here where the X is. And so because we have a double crossover, it inserts the donor DNA, and this, like in humans that doesn't have a centromere, this is destroyed in bacteria. Um, it doesn't stay around long because it thinks it's a virus or something. It doesn't want to have that sitting around inside its nucleus, not nucleus, in its cytoplasm because bacteria don't have a nucleus. So anyway, we this ends up being C minus, B plus, A plus, and it's due to this HFR being able to transfer these genes. All right. So this is just showing you we can have an F prime plasmid um, because that F. So let's go back. So we had F go in, right? And so F can get copied, but it can also jump back out. And so this F prime is a plasmid that contains that when it comes back out, it takes one of the genes with it. So in this instance, let's say that there's a lac plus. This is an insertion sequence. That's what IS means. It's just a short repetitive sequence. And so it's inserting into here. It inserts between the insertion sequences. Now this has become an HFR, right? Because like we said here, when the fertility factor integrates into the main chromosome of the bacteria, it becomes an HFR. So this is just showing you it's inserting next to a lac plus gene in this repetitive sequence, and so um, this is this part C is the HFR that's been integrated. Now it can come back out. It can excise itself. It can flip the chromosome into a loop. It has a crossover here. If you kind of follow that, you'll see that it can take this out. And in this case, when it did take it out and it made another plasmid, so now it's back to this. So it's come out. This red part is no longer in here. It's come back out. But it could take some blue with it. It can take genes that are next to it with it. And so this could be like the lac gene. So there'd no longer be a lac gene in here. It would be on this plasmid. Does that make sense? Um, and we call that an F prime. So an F prime plasmid has the fertility factor plus something else, right? In this case, it's lactose. It's lac plus. It took it with it whenever it excised out. So if you looked at this chromosome of this bacteria, you would have the original chromosome, and then you have a plasmid, and that plasmid would have the fertility factor in it, we'll just call it F, which is a gene, and then it would also have the gene for lac plus, which is an operon, and we're going to talk about that in a minute. Okay, so you could have a transfer of um, a plasmid. So we transfer it into a F minus, which has a lac minus. So essentially, this is what's going on. We have a bacteria. It's got a chromosome. It's an F minus, so it doesn't have a fertility factor. It doesn't have the external plasmid, and maybe its only lac gene is broken, so it's lac minus. We don't know how that happened, right? It could be the promoter is messed up, and we're going to go through the whole lac operon in a minute. But for whatever reason, it can't metabolize lactose, so we call it lac minus. Something's broken, and there's three genes. I'm not going to get too complicated with this. Let's just focus on this. So this thing cannot grow on media. Uh, it can't utilize um, lactose as its sole sugar source. So if you grew it on media that didn't have any sugars but lactose, it would die, and you could tell. So, but let's say here comes along an F prime, which means remember it has the fertility factor, plasmid, remember it's small, couple thousand nucleotides, and then it also excised from another bacteria or maybe itself the lac plus since it was next to it. And then here's this thing's chromosome. So it's not an HFR, it's an F prime because this plasmid is not in its genome, its main DNA. And then so this has the fertility factor, and it can transfer using a pilus. It can transfer that plasmid into uh, a recipient, an F minus. And when it does that, it's going to transfer the lac plus on that plasmid. If the whole plasmid goes across, this would also have the fertility factor, so this would be an F prime as well because now it has both the fertility factor and um, another piece of gene that's functioning, in this case lactose. So now this is temporarily diploid. It has lac plus on an exogenous piece of DNA outside of the main chromosome, um, outside of its genome, exogenous. And then this one is lac minus. So this is actually lac plus and lac minus. Now these aren't linked, they're on a different chromosome. This can replicate independently of this. So that's what we call a partial diploid because it's only diploid for whatever is found in the chromosome and the plasmid. Okay, so I'm not. You guys can watch this. It's basically just a video on how HFRs and F pluses work. I'd have to do a lot of manipulation on the sound of this recording to be able to play that for you. But take a look at it. It's an animation of how this functions. Okay, so we have an F plus, right? F plus means it has the external plasmid that has the fertility factor on it. It can give to an F minus. It's through rolling circle. It's plasmid. So in this case, this one still is an F plus. This original one is still an F plus. It's just transferred its plasmid into here. And now this one is also an F plus. So if you have an F plus and an F minus, they both become F pluses. This is, again, bacterial conjugation. So it's going to make a pilus. We have HFR. Remember, that means that the the F factor is now integrated into the host chromosome, into this bacterial's chromosome. So if it's going to rolling circle replicate, it has to rolling circle the whole 4 million nucleotides. That's never going to happen. So part of this will go into here. Um, Remember, the fertility factor starts in the middle. So it has to go all the way around to get back. So it would have to transfer its whole 4 million nucleotides to go all the way back. So here's, there's only half of the fertility factor here. The other half is left behind on the other side. We start here. Hopefully that animation in YouTube shows you this. So we get we don't get the whole fertility factor, so it's an F minus. And then, we have the crossover event. So uh, we can transfer genes into here from here. This is going to retain all of its original stuff because it's not pushing its own chromosome, it's just pushing a copy. Now we have an F prime. So this one has a plasmid with the red fertility factor on it, but it also has another piece of DNA from either its own uh, genome, right? Its main chromosome, its 4 million nucleotide chromosome. Uh, and this could be like a lactose or three and eight, whatever. It's called an F prime because it has another piece of DNA in there besides the fertility factor that it's taken whenever it excised itself out of the main chromosome. It can conjugate, so anything with an F, any with the F gene in it. So this has an F gene in it. It's in the main chromosome. This has an F gene in it. It's just the only thing in this plasmid. This also has an F gene in it. Uh, that's shown here in red, but it also has another gene which is shown here in blue. So all these have the fertility factor. This one is F plus because it's outside, nothing else. This one's called HFR because it's integrated, and this one's called F prime because there's an exogenous piece of DNA outside of the genome, exogena, whatever. And then, of course, it can make a pilus too using the fertility factor, and it's going to rolling circle replicate this plasmid. It's easy to do the whole plasmid here. And so this F prime and F minus are going to end up making two F primes. And in this case, we're going to have a partial diploid because we would assume would have an original gene. Maybe this is lac plus, and this also could be lac plus. So it's important you understand that because we're going to talk about how we can predict the outcome of what's going on in these partial diploids. So, in order to understand that, we're gonna talk a little bit about lactose. This is really highly studied, and so it's very well known. Lactose metabolism, especially in E. coli. So, lactose is a sugar. It's a disaccharide. This is a glycosidic linkage. It's a beta glycosidic linkage. I don't know if you remember this from 181. I teach my students this. So there's alpha linkages and beta linkages. This is the alpha linkages generally in like what you call carbs, which we call amylose or amylopectin, which on your nutritional label will be carbs. And then this is also in your sugar storage. And then beta would be from things like cellulose, which in your nutritional label would be like undigestible or insoluble fiber. Also chitin, which is like the shells of lobsters and insects. Um, uh, anyway, so and this really has to do with the orientation of the hydroxyl groups. This is probably getting way too deep, but the hydroxyl groups can be cis or trans. So trans is across, cis is on the same side because these bonds can't rotate. So anyway, and then this is glycogen, which is what you store as your carbs, and this is in animals because we mainly store our energy as fats. So so it's easy for us to break alpha bonds. It's not so easy to break beta bonds. So we need uh, a special enzyme to do that, and that enzyme is called beta galactosidase. So some people have this enzyme and they're able to break this beta bond. Some people don't. Some people need bacteria in their intestines to break it down. So like 25% of people have this gene to break this bond, and the other 75% that are either lactose intolerant or they have bacteria in their gut that break it down for them. So this disaccharide, it's way too much information. Just like I said, so this is the trans conformation. So these are made of uh, the beta form of sugars. Okay, enough of that. So these two sugars are galactose and glucose. That's what they're broke. What that's what beta galactosidase does. And if you remember anything from your intro biology class, when you want to break a bond, you have to add water. And when you want to build a bond, you take it out. So adding water is hydrolysis. So this enzyme adds water, breaks this linkage into galactose and glucose. All right. That's all I'm going to say about lactose metabolism. It's probably too much. What I want you to know for the test is that there's a beta galactoside linkage linking this disaccharide together, which we call lactose. That enzyme does uses water, right? So it has R groups that attract water. They're hydrophilic, not hydrophobic. That water comes in and splits this bond and forms two sugars. And you should know from introbiology that usually when you break a bond, you're going to release energy, right? So if you like when you're talking about digesting your food, that's what's going on. You're breaking the bond, you're breaking it down so that you can release the energy from those bonds. All right. Okay. So and the reason that you need to know that is because we want to know about lactose metabolism, and beta galactosidase is one of the enzymes required for lactose metabolism. So this is just showing you that lactose is an inducible expression. That means that if I add lactose to a bacteria, it turns these genes on. And if lactose is absent, then the genes turn off. There's other regulation in that, but basically, if you add lactose, it doesn't take long, and you get synthesis of the enzymes required to break down lactose. That's why it's called inducible expression. Okay, so in bacteria, the bacteria are a little different than um, other eukaryotic organisms. And the way they are is that in most eukaryotes you have a promoter, a coding region, and a terminator, which we talked about, right? So promoter, and then whatever the gene is, and then a terminator. And so, it gets processed. We talked about that already into, an RNA, and that RNA is turned into a protein. So pretty simple, right? One gene, one protein. But bacteria don't do it that way. They do something called operons. So an operon basically is it has a promoter. I'm just going to call that P. And there's also what we call an operator, and we're just going to call that O, which is shown here. And then it's usually like a series of genes that are being made together. It can do this because it has Shine-Dalgarno sequences that we talked about, right? And so the ribosomes can jump on here, recognize the Shine-Dalgarno, and then read that, and then it'll stop at the terminator, or whatever. Now, most eukaryotes can't do that because they start reading at the 5' end um, and continue until it would stop, and then that's it. So that's why we usually just get one gene from one coding region. But bacteria, because of the Shine-Dalgarno sequence we talked about, they can have multiple genes in the same transcript. So this is DNA, it's turned into RNA, and then each of these can be read separately by ribosomes. So in this case, we have something called LAC-Z, which I'm just going to make it a Z. But that stands for beta-galactosidase, which is shown here. Remember, that's the enzyme that breaks that beta bond in lactose to digest it. Then we have something called Y. So this is LAC-Y, and LAC-Y stands for permease. So I don't know if you remember this or not, but in intro biology, you should have learned this, that the cell membrane is selectively permeable or semi-permeable. And that means that only things that are small and uncharged and hydrophobic can pass through this membrane without a channel. The channels that exist in these membranes are very specific for what they pass. So there's a channel for water, and that channel is called an aquaporin. And it's called an aquaporin because it can't transfer anything but water. Otherwise, we just call it a porin. So this is specifically a water hole. It can't transfer iron. It can't transfer lactose. It can't transfer anything but water. And you have lots of these channels, right? So if you want to bring in glucose, you need a channel for glucose. You want to bring in lactose, you need a channel for lactose. This is no different. So permease creates a channel that allows lactose to enter into the cell. And then there's another thing called A, which stands for transacetylase. And I'm going to tell you, nobody knows what the heck this thing does, because if you knock out this gene, it doesn't affect lactose metabolism at all. So you can kind of ignore this gene, like nobody really cares about it. But you should still know that it exists. It's part of the operon and what its name is. But we're not going to do anything about it because whether it's there or not, it doesn't affect metabolism either way. Now, remember, bacteria chromosomes are circular. So this is just showing you it's further away, down upstream. These are connected, but this is an intergenic spacer, an area between genes. This is called LAC-I. Remember, lactose is inducible, which means if we add lactose, it turns it on. So this I stands for inducible, even though it codes for what we call a repressor. Now, normally this is expressed, right? The promoter turns this thing on, it makes a repressor, it recognizes this DNA sequence right here, and it binds to it, right? So I'm just going to fill this in. This is the LAC-I protein that came from the LAC-I gene, which is over here. It also has its own promoter. It isn't controlled by an operator because it's always expressed, so we don't need an O there. But it does have its own promoter. This promoter is generally always on. So this is always being made into an RNA. It's always being made into a protein. And the protein for it recognizes the sequence of DNA and the operator of the LAC operon right before the Z gene, shown here, and it binds to that. And when it binds to it, it physically blocks the polymerase from binding here. So it effectively turns this gene off. So this gene is off, right? It's always off. If you do nothing, the gene is completely 100% not being expressed. That means no LAC-Z RNA is being made, Y, or A. And so we don't get the products of Z, Y, or A. And then this is just another showing you this. So the polymerase, the RNA polymerase, can't physically go across the operator area because LAC-I is blocking it. So we get no gene expression, right? Um, there is some leakiness here, so you're going to get a very little bit of a gene expression. Like sometimes, this will sneak around, but for the most of the time, it's going to almost completely shut this off. And that's where no lactose is present, right? We don't want the gene on unless lactose is present because it would be a waste of energy. When we have lactose present, lactose itself, it's actually metabolite of lactose, but we're just going to keep it simple here. Lactose binds. When we have one molecule bind to another molecule, it changes its shape. You can see this has a different shape. That shape does not recognize this operator anymore. So this can't bind and it cannot block the polymerase. So we get expression of all these genes as one mRNA, right? Shine-Delgarno allows this. So we can get beta-galactosidase, permease, and transacetylase all made in equal amounts from a single mRNA transcript at the same time. And that's how that works. Okay, so basic components of gene regulation in prokaryotes and eukaryotes, transacting factors. So transacting factors mean that the regulatory proteins that normally bind to DNA and regulate expression are diffusible. That means they can go across. That's what trans means. So you could have something on chromosome number 1, like an example would be the lactose thing, and it can affect something far away. Like we have LAC-I here, it can move, right? It can float around in the cell. That protein can float around in the cell and bind anywhere that it finds that operator. That's a transacting factor. Usually these are proteins, right? Because they can float around, they can move around the cabin. We have cis-acting factors. Cis means same. So these can't move, right? They're on the same segment that they're found. So they're pieces of DNA that don't encode proteins generally, and they interact with these protein factors, these transacting factors. So if we were to go back here, we would say this operator is cis. It can't move. It's a part of this DNA, right? This Z, right? The DNA itself is cis. It can't move. This promoter is cis. It can't move. So cis factors are usually DNA sequences. This protein, so LAC-I itself, the DNA, is cis. It can't move anywhere. It's stuck. But this protein is made. Now it can move into the cytoplasm. So that is a transacting factor, and it can go here or anywhere else in the cell. And that's going to be important later. Okay, how is this system discovered? So they identified strains carrying mutations for LAC metabolism. They looked at these partial diploids, these F prime strain, and then they deduce the molecular mechanism behind it. So let's just go on and look at this. Okay, so this is how you actually do it in a lab, right? You're gonna grow these bacteria in a petri dish with supplemented with lactose, right? Um, if it's present, it's going to activate it, and it's going to break down the lactose, right? Z and Y. This opens up the cell. This physically breaks that beta bond, so it breaks that disaccharide. And in the end, the amount of lactose in this plate is going to go down. You don't want that. So this is kind of like a sweetener that's 0 calories. This is called IPTG. It looks like lactose. It activates the lactose genes, but LacZ cannot break it down. So it induces the gene to be turned on, IPTG does, but it doesn't reduce its amount because the genes in the operator can't affect it. It can't break that bond, which is good for researching, right? Because we don't want to have to keep replacing lactose into the media. All right, so these are uh, abnormal systems in in lactose operon mutant strains, and these are just the terms, right? So we say Z minus. That means that it can't make the beta galactosidase doesn't function. Lac Y minus. That means permease does not function. OC means the operator is constitutively expressed, right? So beta galactosidase and permease are always expressed. So what's going on here? Um, we have the promoter, the operator, lac Z, lac Y. I'm not even going to bother with A. Okay, so what happens is this DNA gets mutated. And if this DNA gets mutated, the sequence is not recognizable by the LAC-I protein. So the LAC-I protein cannot bind to the operator. And if LAC-I can't bind to the operator and the promoter is recognized by the polymerase, this is always on. So that's what we mean by constitutive. It's always on. This is because LAC-I can do nothing to shut it down. Doesn't matter if lactose is present or absent. LAC-I cannot bind to this operator because the DNA sequence is mutated. I minus means that I'll just draw it over here. So we have our promoter, operator Z and Y. And then upstream we have a promoter, and then we have I over here. So let's say the protein I, which is the repressor, right? It's called I because it's an inducer. But that protein is made, but it doesn't have the right shape, right? Or so it can't recognize to a normal operator, or maybe the promoter's broken and then this is never made. Regardless, this can't recognize a normal promoter. We're just going to call it O. So that would be a normal promoter, even if it's made, right? So there's nothing wrong with the DNA like here. This would be a constitutive because the DNA is mutated. This is that there's something wrong with this protein. The LAC-I protein is messed up, or maybe it's not made. Can't bind there. So what does that mean? It means both LAC-Z and LAC-Y are always going to be on. Doesn't matter if we add IPTG or not, because there's nothing to shut this thing down. IS means it's a super repressor. So In a super repressor, what happens is these proteins are complicated, right? So let's say this is our lac I, and you know that it it has a binding site for lactose, right? And if lactose binds it, it changes its shape and it can no longer bind here, right? If lactose is absent, it has the normal shape and it binds and shuts this down, right? It's blocking this. I'll make it a diamond. So I is going to block the polymerase, RNA polymerase. Now, in a super repressor, the lactose cannot bind to lac I. And so because it can never bind to it, it can never prevent it from blocking the promoter. So lac I is always here, regardless of whether lactose is present, because lactose cannot bind to it. And that means that it's always going to shut this down. Nothing can be expressed. It doesn't matter if, and remember, IPTG is just another name for lactose. It's just what's used in petri dishes because it can't be broken down. So you don't have to keep adding lactose in it. P minus is just what you think. The promoter is jacked up, and if the promoter is jacked up, the polymerase isn't going to recognize it, and it doesn't matter what happens here. Nothing's going to be made. All right, so those are the possible um, mutations that can occur in this lactose system. All right, so there's some problem sets like this on for the final exam. This isn't on the second exam, on the final exam. So let's say we have a genotype that's O plus Z plus Y plus. So well, let's just draw it out. I mean, I like to do this. So here's our promoter, here's our operator. We know it's right after the promoter. It's not part of the gene. And then we have lac Z and we have lac Y. We don't care about A, so I'm not even going to bother with it. And then over here, further upstream, we have a promoter and then we have I. So I is normally going to be made. And I is normally going to bind here. So we're going to have I being bound there. So if the operator is normal and lac Z is normal and lac Y is normal, so this one isn't mutated. It makes a normal product, beta galactosidase. This makes a normal product, which is permease. This is completely normally functional. It's not constitutive. The sequence of DNA is not screwed up. Lac I, the protein, can bind to it normally, and it's going to block the polymerase, even though it can recognize the promoter. It's going to block the polymerase, right? So if we don't induce it, that means we're not adding lactose. Right? So lactose is not being added. Then we would expect nothing would happen, right? The lac I is going to bind here, block the polymerase, so nothing is going to be expressed. And so we would expect nothing. Lac Z shouldn't be expressed, and lac Y shouldn't be expressed. Now, if we add lactose, remember lactose binds to I, changes its shape so it no longer can recognize this. So this cannot bind. Sorry about that. I'll draw it back. We'll get rid of these two. So the I with the lactose on it cannot bind to the operator. Operator's free and clear. And so in the presence of lactose, this can be made into an RNA. And that, since these are normal, they would get made into a protein. So if we induce it, that means adding lactose or IPTG, which is more commonly used. We should get plus. We should get lac Z made. It's going to be turned into an mRNA and a protein, and this should be plus, right? It's going to also be made. Now, here we have the F prime plasmid, so this is a diploid. So now we have to draw out two. So um, this is saying in the genome, O is normal. We're going to assume the promoter is normal because they didn't tell us it's not. And Z is normal, and so is Y. So this is the genome, the main chromosome, and then we have this F prime. Remember, the F prime is a plasmid, external piece of DNA, and in this plasmid we have an O plus. So I'm not going to write the plus, and we have a Z minus. So Z is broken, the DNA is messed up, and we have a Y plus. Y plus. So this is normal. This one's messed up, but this one's normal. So we have. It's not telling us that LAC-I is present in the plasmid, so we're going to assume it's in the genome. So LAC-I is normally going to be made, right? There's no operator controlling LAC, and so this would be the protein. This would be the trans element. It can move around. It can move here, and it can move here. That's why we call it a trans. Trans means across. Uh, so normally I would bind here, and even though it's not in the plasmid, it still can move, and it would also bind here. So binding there would shut everything down. So in the absence of um, IPTG or lactose, we're going to get negative value, right? Because the operator is normal. If that said something else, if it said minus or constitutive, this wouldn't be the case. But because it's a normal operator, the I protein will bind to it and block the polymerase from turning this into an mRNA. Same thing here. So it has to have a promoter. Without a promoter, it wouldn't be a gene. All right. Now, if we put in uh, lactose, we add the lactose, it's going to bind to this thing, change its shape, and it's no longer going to be able to bind to this operator. So we're going to erase that. We're going to erase that. And these are going to get expressed. So this one is going to get turned into an RNA with Z and Y, and their normal sequence is normal. So we're going to get the protein Z. I'm just going to use circles for proteins, and Y being made. So Z and Y gets made. So this one, Z does not get made because it's red. There's some mutation in here that causes it to not fold in the right shape. So this can't break down, can't break that beta bond in lactose. Y permease is normal. It doesn't matter if you're a heterozygote because this is a heterozygote. So this is Z plus, Z minus. In the heterozygote, you're still going to get Z made. This one is homozygous for Y plus, Y plus. So we're going to get twice as much permease made, but who cares? We only need one amount of permease to get the lactose into the cell. And so we're going to get plus for this, and we're also going to get plus for this. Now, let's look at this one. So this is only on a single chromosome, so I'm going to go over here. We have the promoter, which is normal. It didn't say anything about it. The operator's constitutive. Okay, remember that means the DNA is messed up. And that means the I protein cannot recognize and bind to that. And then this tells us Z is normal, Y is normal. Z is normal, Y is normal. Okay, so if there's notes down here if you guys want to read. So if we have I, it cannot bind to C. It can't block the thing, so we're going to get the mRNA and the protein made. So we're going to get Z and Y even if lactose is absent. So we should get a plus non-induced plus non-induced for both Z and Y because they're both normal. Again, we add lactose, it'll bind, all right? And it will change the shape. But this still can't recognize that sequence, so it can't shut it down. So we should get plus, because it can't, this can never bind and block the RNA polymerase from binding to the promoter and reading those genes. All right, so I'm going to erase the first one, and we'll do this other problem. Okay, this one is, so it's a diploid, right? It has the F prime plasmid. So in the chromosome, we have a normal promoter. That didn't tell us anything about that. A normal operator. We have Z minus, so this one's broken. Sorry, minus. This one is Y plus, so that one's normal. So we're never going to get Z made. No Z ever here. Y is going to be under the control of the operator. And then we have the lac I. So we're just going to make that a protein, right? Without lactose, it's going to bind to that operator and shut this whole thing down. But we have another plasmid. So this is in the genome. This is the other plasmid. This is, dang it, this is the F prime because it has part of this gene in it. So our operator is constitutive. Our Z is positive, so that means it works. Y is broken, so we're never going to get Y. It doesn't matter if this is on or off. Y is done. And then we have a normal promoter. So that means that this lac I cannot recognize and bind to this ever. So this is always on, no matter what. So we're going to get Z made from this plasmid all the time. And we're never going to get Y made from this plasmid, even though the gene is always on. So Z is going to be plus plus, right? 100%. Because we're going to make it from this plasmid no matter what. Now let's look at this one. Z, we're not going to get any Z from this. So that would be minus, but we have the plasmid that is always on, so it's plus. Now, this lac I works. It can't bind to this. It can't control it, but it can't control this one. And so if we have lactose present, it's going to bind here. So normally, without lactose, this would be shut off. So we're going to get no Y. We're going to get no Y no matter what happens, right? It's even though this is always on, Y is broken. This one is under normal control. So if there's no lactose, this is going to bind and shut this down too. So it's going to be Y minus minus. So we should get no lactose, no Y. That's going to be different. This is going to be always on. We're never going to get Y from this. So we don't care about this for Y. But when we have lactose present, this lac I is going to bind to it. It cannot bind to the operator. Its shape isn't right. And so this is going to get turned on, and we're going to get Y made. We're going to get Z made, but Z is broken, so it's not, this doesn't count. And so in the presence of lactose, when it's induced, we're going to get Y plus. And that's what that's telling you. Okay, so I'm just going to kind of wrap this up. I don't want to go much longer than an hour and 15 minutes, but so operators are cis-acting, right? That makes sense. They're DNA, right? We said DNA is a cis-acting element. So these operators, they're not going anywhere. They're part of the DNA. Operator specific for the DNA sequence that it binds to, right? If we get mutations in here, then the lac I is not going to recognize and bind to that sequence. I'll let you guys go through these. We kind of talked about this, so I plus or minus Z or whatever, and then you can do experiments on that. But just so you know, I plus, I minus. So I minus means that the repressor's not being made. It could be the promoter's broken. it could be um, it could be that it's not folding in the correct shape. But for whatever reason, we're not getting, so I minus means that we're not getting a repressor. I plus means we're getting a repressor. Remember, this is a trans element, so it can go anywhere, like the example I just gave you. And again, I'm going to have you guys do these on your own. I'm not going to go over that again. Uh, the super repressor, we talked about that. Remember, super repressor, the lactose or IPTG can't bind. So this is always going to bind to this. It's always going to be off. Again, same thing. Here, super repressor uh, is not going to be able to bind to it, this super repressor. Now, this is I plus. This is normal. So this will bind and shut this off, or not be able to bind to the operator, right? So this I plus is normal because it's a trans element. It can also work over here. So even though this is a super repressor, this one can go over here and shut and affect this. So you got to think about when you're doing these problems about these trans elements that can go, they can move around, right? Um, I think I'm going to stop here at the positive control, the lac operon, and um, let's just do a super repressor one real quick, and then I'll wrap this up, and I'll let you guys do the other ones. So let's do this one. This one looks like it's a little bit hard. So we have the genome has the I super repressor in it. Remember, it has a promoter, and then we have lac Z and Y. It's not telling us anything about the operator, so we're going to assume that the operator's normal and the promoter's normal. It didn't say if it's not or is or not. So in this case, the super repressor is always going to bind here and shut it off. It doesn't matter if we have lac, also IPTG, or not. This is always going to shut down this because this protein does not bind lac, so it can't prevent it from binding to the operator. And if it's on the operator, the promoter can't. So this whole gene is always off, right? Now, we also have a plasmid and F plasmid. So this is the plasmid, and it has I plus, so it's a normal I, normal promoter. This is a normal promoter, normal operator, and then we have Z plus and Y plus. Okay, so this thing is going to bind to this operator. This thing is also going to bind to this operator, right? So even though we have a normal I, right, this normal I would also bind, but if we have lac, which is IPTG, same thing, it would bind to this and it couldn't bind to the operator, so this would be on. But this super repressor, even though it's in the chromosome, can also bind to this operator and shut this off. So this is always going to be off because lactose can't control this super repressor. It's a trans element, and it can go here and park itself here, and it can go here and park itself here. And it doesn't matter if lactose is present or absent. These are both going to be shut off. So we would get minus for everything. So I want you guys to practice some of these. And if you have any questions or whatever, like I said, I have office hours every day except for Monday, and no one ever comes. I mean, it's super rare. So if you need help, come to my office hours. I'm the best tutor for this class. I make the tests. Um, and or send me an email if you can't make it to office hours. But anyway, so that's the bacterial genetics. That should cover most of that stuff. We're gonna next time we'll talk about the positive control of the lac operon. Actually, maybe I can just finish this. Um, okay, yeah. So there's something called cyclic adenosine monophosphate, which is CAMP. So the lac operon is controlled by lactose, right? Present or absence. But it's also controlled by levels of glucose, right? Because if you have glucose as an energy source, it's a you don't have to break glucose down anymore. It's a monosaccharide. So you would preferentially use glucose over lactose uh, anytime. And so if there are high levels of glucose, uh, we don't. Uh, okay, I'm going to finish this. So glucose regulate levels or regulate cyclic AMP levels. So when you have high glucose, then uh, you don't get uh, high levels of cyclic AMP produced. And so there's no cyclic AMP in situations where you have high glucose levels. No cyclic AMP is made. When in low glucose levels, you get high levels of cyclic AMP. Um, so basically, what this is showing is in high glucose, you're making more ATP, and higher ATP affects uh, the production of cyclic AMP. And in low glucose, the ATP levels are low, and so you get higher levels of cyclic AMP. So anyway, and let's just forget about ATP's role in this and just say high glucose equals no cyclic AMP, low glucose equals high levels of cyclic AMP. Now, in the promoter region, there's a protein called CAP that binds to cyclic AMP, and this is a transcription factor that helps recruit the RNA polymerase. So, if these two are present, we get an increase in gene expression. If these two are absent, we get a decrease in gene expression. So, this makes sense. If you have low cyclic AMP, that means you have high glucose. So, if we don't have cyclic AMP, it can't bind and it can't recruit the polymerase. So this gets shut off in high levels of glucose because we don't want genes to break down lactose if a lot of glucose is present. In low levels of glucose, we get a lot of cyclic AMP. So that we get this transcription factor that recruits the RNA polymerase, that converts the DNA into RNA, because in low levels of glucose, we want to use lactose preferentially. So essentially, that's how that works. Let's see, what else we got? So glucose is present, cyclic AMP is low, no lactose, no lac mRNA. Simple. Um, when glucose is present, cyclic AMP is low. So, and then lactose is present. We want to get, since glucose is present, we don't need to make the genes to break down lactose. We're going to preferentially choose glucose. In this one, if glucose is present but there's no lactose, then there's no reason to make any uh, genes that break down lactose. And if no glucose is present, that means cyclic AMP levels are high. Then this camp is going to recruit the polymerase here, and the gene expression is going to be turned on. So we're going to get a lot of lac mRNA. Remember, the lactose is binding to this, so it's not going to block the operator as well. So it's basically two feedbacks. We have the I, which is the repressible system, and then this, which is the inducible system, right? If it's present, cyclic AMP is present, then it induces expression of this gene. All right, so that is the lac operon in a nutshell, and I will see you next time.